What I was going over in that last lecture was in this graph here showing you the generation of an action potential graphically. So notice down here it's minus 70 millivolts. Don't worry about the magnitude, but the minus means the negative charge along the inside of the plasma membrane, okay, of the axon. So with the stimulus, and as sodium starts to rush in, sodium's positively charged because the opening of the sodium protein channels, the depolarization starts to occur. The negative charge along the inside of the plasma membrane starts to get less negative, starts to go towards zero charge. When it hits a certain value called the threshold, then it spikes, meaning a bunch of sodium rushes in, and the negative charge internally becomes actually positive. And that's what's happened here. And that spike and change, reversal of the resting membrane potential, where it's now positive inside, negative outside, is referred to as an action potential. Okay? And we'll see next that that action potential, this change, reversal in resting membrane potential, propagates down the axon rapidly. So the action potential propagates, as this little video shows. Initially, you have a resting membrane potential. It changes polarity, meaning the action potential happens, and the action potential moves along the axon rapidly. Because what happens is when an action potential occurs in one spot on the membrane, it opens, it stimulates the next spot on the membrane to open up sodium channels, and then sodium starts to rush in there. That area gets depolarized, hits threshold, action potential occurs there. When that occurs there, the next patch gets depolarized because sodium channels open, sodium starts to rush in, hits threshold, lots of sodium rushes in, reversal of the resting membrane potential, and so forth and so forth and so forth, all along the axon. So that's the propagation, the movement of the action potential along the axon as we see here, and it occurs very rapidly. With a myelinated axon, myelinated by Schwann cells in the peripheral nervous system, the speed of conduction is accelerated. So the action potential propagates, moves faster in myelinated axons versus those unmyelinated axons. So like this little video demonstrates, the action potential moves much faster in a myelinated axon versus an unmyelinated axon. It still moves rapidly in an unmyelinated axon, but far faster or much faster in a myelinated axon. Okay, let's look at some organization of nervous tissue in the nervous system, some definitions, peripheral nervous system, and central nervous system. So a group of cell bodies, meaning cell bodies of neurons at the nucleus. That's the cell body, and that would be its axonal extension, but groupings, clusters of cell bodies in the central nervous system is referred to as the cortex or cerebral cortex, that outer layer of the brain, and also nucleus, plural meaning nuclei. And maybe not the best term they picked, because nucleus also means the nucleus of a cell. But nucleus or nuclei within the central nervous system, here darker in color, nuclei, plural, those are clusters of cell bodies in the central nervous system. And we'll see in the spinal cord, also part of the central nervous system, excuse me, there's also clusters of gray matter internally in the spinal cord. Okay? In the peripheral nervous system, like in nerves, groups of cell bodies, clusters of cell bodies, are referred to as a ganglion, singular, ganglia is plural. And we saw that with what's called the dorsal root ganglion, clusters of cell bodies of sensory cells in the afferent pathway or sensory pathway. Bundles of axons or axonal fibers are called nerves in the peripheral nervous system. So the nerves that leave the spinal cord, spinal nerves, or nerves that leave the brain, cranial nerves, to innervate the head, sensory receptors, and the body. In the central nervous system, axonal tracts, well, bundles of axons or fibers are called tracts, or sometimes axonal tracts. So that's what you see here, color-coded in blue and red. Red indicating sensory afferent tracts, blue motor or efferent tracts. So that's this grouping of lighter material called white matter, and the darker material is called gray matter. Okay? So the gray matter is darker because it's composed of cell bodies, dendrites, and mostly unmyelinated axon that just give that tissue a darker appearance. The white matter is lighter in color, not really white, but lighter in color, and much of that material is composed of myelinated axons, and the material, the myelin, is a lipid, fatty type material which has a lighter appearance and color, which is why it's referred to as white matter. This figure 13.6 in your textbook shows gray matter in the central nervous system, a whole bunch of cell bodies, and associated neuroglial cells, a little myelination, but most of this is unmyelinated tissue. And then the white matter, you have cell bodies and neuroglial cells, but a great abundance of myelinated axons, giving it a lighter color, lighter appearance than the darker gray matter. Here's a coronal frontal section of a human brain, and you see that the dark matter is external, the cerebral cortex, groupings, aggregations of cell bodies, and then internally, deeper, there's white matter. So those are the mostly unmyelinated axons. I'm sorry, myelinated axons, which is why it's whiter in color. Okay? And those would be tracts connecting regions of the brain. And notice here there's clusters of darker matter internally, deep. Those would be nuclei. Okay? Clusters of cell bodies. Gray matter. Okay, that's it for this chapter.